I am Mr. Ilando, Principal Technical Officer, ICMA DCRC, Pondicherry. Today I am going to take about the tick and the Fleas biology and the control. I think most of you who are entomologists, you would have studied about the mosquitoes. But this is a totally different. But these ticks and fleas are transmitting various types of disease in the last 2 decades. Why we should study the tick-borne disease? The tick-borne diseases are second to mosquito-borne disease. Among the arthropod vectors like mosquitoes, ticks harbor the largest diversity of microorganisms, including virus, bacteria, babushia, and the finally nematode also. But mosquitoes transmitting only a very few. But apart from this, the tick is transmitting all type of microorganisms which transmit to the human. The number of infectious disease in humans has increased. Approximately 335 human infections have emerged since 1940. How many diseases has been identified after 1940? And approximately 60% of the diseases are zoonotic. Zoonotic means the parasite transmit from the animal to the human. Okay, the parasite transmit from the animal to the human. It's a zoonotic. Of which 72 are transmitted from the wildlife, and 28% are transmitted from the domestic animal which is living surrounding us. Furthermore, approximately 30% of the emerging infectious disease are vector-borne, which include tick-borne disease. So this is the overall vector-borne disease. You see the mosquito, mites, day fleas, sandflies, tsetse flies, lice, fleas, red bug, and the ticks. And the ticks, if you see there are how many type of diseases transmitted to the human from domestic animal to the human. But most of the diseases are reported in the foreign country like USA, Russia, and other things. That the Lyme disease is very famous disease. Rocky Mountain spotted fever also, it is very famous in America. But in India, the main disease is KFD, which is present in the Karnataka state. This is present in the Karnataka state. The next one is the prevalent tamaric fever. This is also one of the outbreaks taking place in the around Gujarat state. Then these are small virus, conjunct virus, bubble virus, and apart from this, there's recently Indian tick types also transmitted from tick to the human. And what are the common big diseases present in the world? This already I explained to you. We will see what are the main diseases present in India, which is what are the vectors and this present. For example, Kaiser's forest disease, it is transmitted by the Hemophilis spinifera and the Hemoviruses futuris. This is the tick which is present in the Shimoga district. Then CCHF, that is a criminal common hemorrhagic fever, which is transmitted mainly by the Hyaloma anatolicum ticks. Then Borindia burkafori, this is the Lyme disease. This Lyme disease mostly present in the foresting area, and it is transmitted by the Ixodes sicilis. Then recently, last 5 years, Rickettsia cholerae indica, which is the very specific present only in India only. So that's why it is called the Indian tick typhus. Now, using the diagnostic method, now people are finding many states they are reporting this Indian tick typhus. This disease actually transmitted from the dog ticks to the human. So the main vector here is the rhizocephalus sanguinis. Then another is a conjunct virus, which is transmitted by the human cellular intermedia, which is not very dangerous disease, but this also present in the transmitted disease in India. And so before starting this, we just have some morphological feature of the ticks. The ticks having let's say 3 regions, but it cannot be deciduous like mosquitoes. This is the head. This is a head region. This head region usually called the hypostoma. The hypostoma, this is the basis capital A. So this is the hypostoma, which actually penetrates to the skin of the animals or human, which is helpful to take in the blood from the domestic animal. We will see the detailed morphology about the state flies.
இல்லை சார், அவர் வேற யார்ட்டையும் பேசிட்டு இருக்காரு, நீங்க கண்டினியூ பண்ணுங்க சார்.
Okay. This is we see the ventral and the dorsal view of the ticks. If you see that the special of the ticks is in most of the insect or the animal, usually the genital part is present in the posterior region of the body. This is universal in most of the insect and the most of the animal also. But for the ticks, the genital region is present just nearer to the mouth part. This is totally different from the other animal kingdom. And you see that how can we difference between the male and the female? The female they have scutums is present. This is called the underscutum, but this scutum is absent in the male tick. If you see the darker slide, this scutum will be absent. So this is a hypostoma, which is helpful in the piercing the skin of the gem, and these are the underscutum. And also you can see there is a rectangular part called question, which is very much helpful for addition of genus level. And you see, this is the actually male tick. You can see that the male tick they have a genital aperture here just nearer the mouth. Usually, this genital aperture present with the first coxa and the second coxa. But as far as the female, the genital aperture present between the second coxa and the third coxa. So usually the ticks have the 4 pairs of legs, but in the larval stage they have only 3 pairs of legs. So the 4 pairs of legs, what are the things? This is the first leg, second leg, third leg, and the fourth leg. So each one they have their certain function. And if you see the digestive system of the ticks, you can see that they are very much diverticular is present here. That's why when you see the ticks while on the dog, you might have seen that it would have been very bulged. It is more than 10 and 20 times the bigger than the normal unfed one. Because this is mainly due to the diverticular present in the ticks. And the first we see now the tick life cycle. That after mating, is usually the mating will be taking place outside the host, sometimes on the host. So most 95% of the mating taking place on the coast only. So after mating, there is a process called the female lay the egg. Just to see, you can able to see that large number of eggs which is laid and it will go up to the above the dorsal side. And after 10 days, the egg will be hatched with a small larvae. That is, they have only 3 pairs of legs. Then after that, this larva, this is the highlighted one, this enlarged one. So this is have only 3 pairs of legs. They have the larvae. What is the another specialty of the tick is the any type of insect without food they cannot live for a long time. But as far as the tick is concerned, these were all the same. The larva, nymph, and even others also. They can without food they can live up to even 200 days. So that is a very specialty of the ticks. Then this larva will become nymph after feeding on the host. Then the nymph will become adult after feeding on the host. Then finally, this will become mate. Then the cycle will be continued. So just to take how that life cycle is taking place, apart from egg to larva to nymph to adult, there are different type of post cycles. What is the one post thing? One post thing means the larva gets to enter into the domestic animal. Then the larva become nymph in the same animal. After that, the nymph also become adult in the same animal. So they will not fall down when the larva enter the domestic animal. So the larva, until become adult, it will be reside only in the domestic animal. After they become adult, they take the blood, then only they will be falling on the ground for laying the egg purpose only. So this is called one post stick. This is totally present in the one tick called blue binders. Then another is a two post stick. With the two post stick, the larva which enter into the animal, then in the animal itself, the larva become nymph. So the both larva and the nymph it will develop in the domestic animal itself. But the nymph will fall down to the ground and it will be taking another host. So that's why they called two host thing. So other thing going to the another host and the larvae name developing this same host. So this is called the two host thing. The very good example is called Phylema genus of the pigs. The third one is the three host thing. That is larvae enter the animals, feed it, fall down, and again they burst into limb. Again they go to the another host. They feed on, and then after that they fall down, and again they will develop with the alert in the ground. Then feed on the third host. So each larvae, nymph, and the adult, they are seeking a different host. So that's why it's called a three host life cycle. The good example is Ema studies India. For example, you see the right, this is the right reversible standard is a lifespan. You see that larvae, how many days without a food, they can't be able to live that 24 to 253 days. But after engagement, they can more than 5 days, 6 days, they can be able to take birth. After that, the molding will be taking place. Similarly, see the dream stage, they can unbirth, they can live up to 183 days. So that is the specialty of the deep. Similarly, the adult also, they can able to live without food for more than 80 days also. So the maximum deposited will be 1,600 by a repeatable family, which is you have been knowing very common take present in the dog. That is called repeatable family. And you can see that you can see the pigs present in the almost from the amphibians to the mammals. From amphibians to mammals. Amphibians means from the, even the pigs also present in the frogs also. You can see these are the animal that they part of the pigs, which is mostly present in this is where we collected in India and we reported the pigs. And you can also see that some of the pigs also present in the rhinoceros, elephant also, some pigs are there, tortoise also pigs are there, frogs also pigs are there, even birds, lizards, and snakes. Snakes, they have a special type of pig which is present only in the snake, that the genus called Amonima. So there are two different pigs are present. One is a hard pig, another one is a soft pig. Hard pigs, usually the scrotum will be very larger and the temperature will be visible if you see from the top of the pigs. But in case of soft pig, the soft pig, if you see from the top of the side, the mouth parts will not be visible. So this is a major difference between the soft pig and the hard pigs. And the soft pig mostly present in the birds, fowls, and other things. The hard pigs are mostly present in the domesticated animals. And this is the how to collect the ticks from the ground. So this is the one of the method which usually they do in Kaisno forest. This is where the Shimaga they regularly do it. This is called flagging. Usually the leaves will be present in the forest floor, in the dried leaves or in the grass. So during the coastal period, when you if you flagging the cloth, your leaf cloth over the surface of the forest, then the leaves will be attached to the this leaf cloth. Then this will be taken by the forceps. So this is the regular survey that do the in the Simaga district. Apart from the names, and also we can collect the things from the domestic animal. Domestic animal, you see that the things will be present in the ear side and the abdomen, tail, and the anus. And some of the things present mainly in the anus, that is mostly the hilama thing present in the anus side. Some of the things will be present in the tails of the domestic animal. So these are the collection of the things from the domestic animal. Apart from this, there are another method called collection of ticks from the plants. That is the on the procedure ticks we can collect from the plants. That also so that will be appear mostly after the June up to the December. And if you see the seasonality of the ticks, then we see that the ticks main stage will be mostly present with the January to this January to May, but the alert will be in July to December only. You see, there is a repeatable song is Barsa, Pyramid, this is the one of the study. They showed us how the tick density is increased from March to August, then there will be slow down. And how to calculate the tick survey? What is the infestation prevalence? The number of infected animals divided by total number of animals examined in the 100 is called infestation of the prevalence. Then the tick abundance, the number of collected ticks divided by number of examined animals, both the positive, if you take consideration of both the positive and also negative. Infestation of intensity, number of collected ticks divided by number of infested animals only in positive animals only. So this is the common calculation they are doing for the tick survey. This is the one slide I show you that longevity of the isolated ticks. This is why I am showing this. See, each is the larvae and the game and the adult. They how many days they can able to live without food. That is the specialty of them. For example, Ambulema americana, they can able to 150 days during summer and 290 days, 97 into the winter. So like that, you see that dim after day summer 476 days and the winter also 476. Like this is the each genus they have different lifetime. So if they can able to live without food even for 1 year. So after 1 year, if give food, they will take and be able to survive. This is the reproductive capacity of this species. For example, these are different genus, Amalema, Dermosunda, and the Margoporus. So they have how much maximum egg they can able to lay. They can average, they can able to lay 3,000, even up to 4,000, 5,000 eggs, which is the Dermosunda. Even the last record is around 20,000 eggs also can be laid by the tick species. This is the engagement period. Engagement when how many days the ticks can able to take blood from a domestic animal. Usually there are 3 to 5 days or 4 to 5 days, depending upon the if not disturbing, they can able to take. For example, the molding period they can able to take 10 days or 15 days or 26 days, depending upon the favorable environmental conditions. And this is the life cycle of Spinothera under laboratory condition and ambient temperature. This already I explained to you. This is the only show a single species how the different days of pre-oviposition, oviposition, and incubation differ, but they can be able to live even for 1 year. This is the first Spinothera. There is a Chemophysalis Spinothera. Is the Caisinophorus disease vector. So this is the specialty of Caisinophorus disease, but the names will be present from the January to May. So during that time only, if the person enter into the forest, they may get exposed to the nymphs by tick. You see, the nymphs are the infected stage. If you bite if the infected nymphs bite on the human may get the KFD. And you see that soon in the June to September, the alert will be developed, and from the October to December, the larva will be there mostly. This is the Sintra player from universal stream, which is studied in the Semacatastics. You can see able to see Spinothera, Tuchteris, Caisinophorus, Kinari. These are all the vector. Caisinophorus is vector, which is how the density is increasing up to July. After that, there will not be any next stage. This is the physical prevalence from Hemophysalis order in the Semacatastics. You can see after May, there is an increase in the adult density. Okay, already I told you, I already showed the first slide is that there are how ticks are around 40% of the ticks only present with the domestic animal. Remaining 60% of the ticks are present with the wild animal. So they are apart from the dog, tortoise, wild boar, yak, monkey, camel, tiger, horse. So all type of but the type of tick species are different from one to another. For example, take a snake here. Here the only Abodima genus only is present. And the cost difference. The cost difference, if you see that how that these each and everything have the different cost difference. Spinothera, it is present. It can take the blood from the carpet buffalo, sheep, and birds. But you see, the Kigari, they take only tiger, and the Kaisuru, they take only wild duck. So this is the first preference of the different hemophagus species, which is recorded in the Karnataka state. Then how the tick is controlled. Usually the tick control is taken by the removing the tick from the domestic animal. That is the common method which is applied. Apart because the ticks, if you see that the ticks present very widely in the forest, we cannot spray an insecticide throughout the forest, and also if we apply insecticide in the forest, sometimes beneficial insect and other things will be getting destroyed. So the food chain will be changed. So wherever there is a monkey death is taking place in the forest, around the 50 meter, 50 feet, they will be using more than 25% powder only. So apart from this, some places they are using the fire. So the footpath is they are putting fire so that the larva, the nail, everything will be killed because which will be present in the mainly in the grass and other land. And then these are the bifid-3, corporate, cyclo-3, beta-cyclo-3. These are all the insecticide. Apart from the lot of insecticide present, whatever insecticide is used for killing the insect and the mosquito also can be used for the tick control. But generally, people are not using it because it is the ticks percentage which is present in the very larger area. The application is very difficult. Now let's come to the Flea. So far I completed about the tick and biology and how to control. Now come to the Flea. Flea is what is the main important. That if there are 3,000 species of fleas belonging to about 200 genera in the world, but only few are important and the best is man. In India, around 46 species and the 5 species accommodated 24 genera and the 8 families. About 91% non-flea species are bite mammals and the remainder are parasites of the birds. So this is coming at the order of Cyberaptera, one of the major important, medically important fleas. There are called Eplicidae, which include genera called Girafilla, Cavis, Murex, Tunda, and the genus Amaryllis. Girafilla, Cavis, Austria, and the Basilicus, these are all mostly present in the rodent, which is called the vector of the plague. And the another genus Amaryllis species, which is the genus Amaryllis canis, genus Amaryllis. This is a dog, and a cat, fleas, and which is mostly present in the domestic animal like a cat, even the sheep, and the bird also is flea is present. So this is the common fleas present in the world, and this is the dark flea, which is the genus Amaryllis. Genus Amaryllis is cat flea, genus Amaryllis fleas. So we cannot say that dark flea present only dark only. Sometimes the dark flea also present the sheep and the goat. Similarly, the cat flea is also present in the other domestic animal. So these are the hen flea, the fleas which is present the hen and the, and this is the one called a Pulisidae and which is present as yet it's called a human flea. So what are the symptoms of the flea biting? You can see that angle of the human. You can see the angle of the human where the lot of fleas bite is there. And how can identify whether the dogs catch the fleas? They usually see that the dogs will be sitting and the scratching is their nose side. So we can be able to identify so the dog is infected with fleas. Similarly, if a cat, there is also there is scratching over the head, so we can find out these cats also they have infected with the fleas. And I see just to tell you overall morphology of the fleas. This is called the flea cabin, the head, thorax, and the abdomen region. But just like the face, they are all pieced together. You see the head region, they are in the triangle shape, and also the backside, there is a thorax region, prothorax, mesothorax, atathorax is present, and apart from this, you see that the wet region, there will be a genal form, which is called protagonal form, which is may or may not present. In Gynacilla, both are absent. Apart from this, we can able to differentiate the male and the female by seeing that there is a PDC present. I showed the text slide whether it's present or not. So this is the male. You can say this is a male fleas and the female fleas. Because the female fleas, we can able to see by seeing the spermatheca present in the abdomen, and the male fleas, we can able to see their penis here. So this is the general I told about the external marble. I don't want to go very detailed about the morphological feature because they are already explained about the different feature, and there are different details present which is very much helpful for the taxonomic identifications. So now come to the life cycle of the Flea. The Flea after mating, they will lay the egg. The egg is usually laid near the where the host is present. For example, the Flea which is living on the rodents, which lay egg nearer to the rodent holes only. So they'll be spreading there only. So after that, the Flea eggs will be just like the white color or maybe a yellow color. So the egg will be laid down near the rest of the host. Then after that, the eggs will be hatched within 5 to 11 days. So within 5 to 11 days, the egg is hatched into the larva. The larva may consist of a larva 1, there is a 3 stages, the larva 1, larva 2, and the larva 3. After the larva, they become the pupa. The pupa usually be the same inside the cocoon. Then after if the favorable condition is present, then the adult will be emerging from the cocoon. So we see that both sexes of Flea take that milk, therefore they are actually a vector. The female Flea leave the host to deposit their eggs in debris that accommodate the host delivery, such as the rodent flowers or the base. Eggs are very small in are oval shape, white or yellowish in color. They are thinly coated with the sticky substance, which usually result in them becoming covered with dirt and the debris. That's why when you see that the eggs and the people also, there may be a lot of small sand particles will be attached to this. So that is mainly due to the sticky substance which is present in the egg as well as during the pupal formation. Egg hatch within about 2 to 14 days, depending on the species of the Flea, the temperature and the humidity. A minute legless larvae emerge from the egg. So that's why we can be able to differentiate from other arthropods. The arthropods, some of the larvae, they have the egg legs, but here there is no leg like mosquitoes. How can we differentiate between the larva of the other arthropods and the Flea larva? The Flea larva, we can be able to see there is a 2 finger-like projection in the posterior region. So a small blackish head with a very small pair of antennae will be present, followed by a pale cloud distinct and more or less similar segment. So the first 3 units are the thorax. The first 3 thorax region, each segment bear a circular seed tail near the posterior border. The last segment is ends in a pair of rendered finger-like classes termed the honor stern. The presence of this stern combined with the seed tail on the body distinguishes larval flea from all other type of insect of medical importance. So larvae usually feed on the organic debris included in the host pieces. Apart from this, the fleas, adult fleas, they will take up blood from the coast and they will have excreted. So that digested blood and the excreted blood also be taken by the fleas larvae. Some species larvae are scavengers and they feed on small dead insect or even the dead adult fleas of their own kind. There are usually 3 isolated some species they have 2 insert and some species they have 3 larval insert. But in some species there are only 1 insert only. The larval period they last 10 to 20 days, may be prolonged more than 200 days. by unfavorable conditions such as limited food supply and the low temperature. So this is common for every type of insect. If it is a lower temperature, the growth rate will be very much reduced. Maxilla range about 5 kilometers long. At the end of the larval period, the larva sleeps as a whitish cocoon, as I told you, from silk produced by the larval salivary gland, just like our silkworm. Because the sticky nature, the fine particles of blood, organic debris, and sand picked up from the floor to the host floor. About the 2 to 3 days after having sperm, a cocoon around itself, the larvae pupae within the cocoon. Adults emerge from the pupa after 7 to 4 days. After having emerged from the pupa, the adult requires a stimulus, usually vibration such as caused by the movements of the course within the course, burrow, ability to escape from the cocoon. So these are the stimulants. These are the stimulants which stimulate the cocoon, which is so that the larvae will come out. However, animal sector or houses are located that adult fly fails to escape from the cocoon until their dwelling places are reoccupied. That's why each houses where suddenly if the houses are vacant for a longer time, if suddenly people enter to that particular house, you can see a lot of fleas by biting in the air because suddenly most of the of all the cocoons, the adults will be coming out from the cocoons and they started to biting because they are very hungry. They started to biting. Overall present in this. So the another factor is some species carbon dioxide also emitted from the cocoons, which stimulate the adult coming from the cocoons. Adult may remain alive in the cocoons for about 1 year. If it's not present, so they can able to live in the 1 year inside the cocoon. That's why the lifetime will be very long. This explains why people moving to buildings that have been vacated for many months may be suddenly attacked by large number of very bloodthirsty fleas. So these being newly emergent adults seeking their first blood meal. Other host players move either by rapidly curving or by jumping. Usually the fleas they doesn't have many wings and they move from one place to another place by jumping. So that's why the hind legs are modified in such a way, just like a grasshopper, the fleas can be jumped 18 centimeters vertically and 35 centimeters horizontally. Fleas frequently bite people mostly on the angle of the legs, but the night sleeping person is bitten on the other body also. Okay, now come to the control method. You see, just like a ticks, for here also there are a lot of insecticide they are using for the other insecticide also can be used for the flea control. The flea control naturally so this is a common magnesium, carbohydrate, DDP, pyrethrum. These are mostly commonly used for the pet animals, particularly the dog and the cat. So that they are giving this insecticide and spray over the body and they go to kill the fleas. So clear couples are not very susceptible in this insect. Consequently, insected treatment should be repeated every about 2 weeks about or about 6 months. The reason is the insecticide which you are taking are controlled by the larvae or the adult. So it will not kill the cocoon. The cocoon will be for living for a long time. So during the favorable condition, again the cocoon will come into the adult then they make a again make a biting on it. So for controlling purpose, the insecticide is not susceptible to the cocoons. So in many parts of the world, a gene of select there is a flaky vector and the prevalence irritants have developed a resistance to the DDT, HC, and the diazine. In such cases, organophosphate or concentrated insecticides such as 2% DSP, 2% pention, and 5% malathane 2% panitia can be used. In India, DMs has developed a resistance to the malaria, and recent trial has showed carbaryl to be a most effective insect. Though these insecticides they are applying onto the domestic kind of other things, but the problem is most of the fleas are in the rodents. The rodents are living in the domestic, the peridomestic region, so that is very difficult to control unless you put the rodent inside to kill the rodent. So after that, if the host is pliable, again the flea started to attack the humans. So thank you.